Can you hear me in the back okay? Oh yeah, no problem. We'll get started in about 30 seconds. For those of you who were here earlier, music selections. First one was photosynthesis. It's obvious why I chose that one. That was your last two lectures, photosynthesis. Students tend to do very poorly on the photosynthesis part of the exam. I'm going to go over a few aspects of it where students typically tend to have issues with the exam in terms of their grade. I've also tried to go through common questions during office hours. That was the focus of the slides I put together. Pinball Wizard. The purpose of that song was to, of course, if you've ever played pinball, the ball's up at the top, it eventually comes down due to potential energy. But ultimately, no matter how many times it keeps bouncing back and forth, it will eventually go to the bottom. So the same is true for the antenna. Wherever that photon is captured, that energy eventually resonates to the reaction center where charge separation occurs. The last song, Spill the Wine, was to try to introduce to you what happens if you have pyruvate from glycolysis and there is no oxygen present. You could either form lactate so that you can allow glycolysis to continue, that regenerates the NAD+, or in those organisms that are capable of it, you can make alcohol. Now, we do not have the capability to make alcohol from pyruvate. If we did, if you thought about it, on Friday night, instead of going to the party, you just run up and down the stairs about 20 times, and you get your alcohol. Might be cheaper, but it doesn't work that way, okay? So let's start with a few slides here. If I could have the computer, please. All right. Protein import. Clearly, the destination of the protein is important. You have to have it going to the correct place. So in all cases, without an exception, okay, we're going to be talking about starting with the, we're not talking about ribosomes within the chloroplast. We're not talking about ribosomes within the mitochondria. I'm going to be focused on ribosomes in the cytoplasm. You know that the subunits are assembled where? In the nucleolus. Where is the ribosomal RNA made? In the nucleus, in particular at the nucleolus. So that's where the ribosomal RNA is made from specific genes that encode for it. The proteins that correspond to the proteins within ribosomes are synthesized out in the cytosol. Okay? And then they have to be imported into the nucleus. What protein is responsible for import of proteins into the nucleus? Important, okay? So in all cases, let's start with what happens. From the DNA, we get the mRNA, because we're going to be talking about protein. That mRNA gets transported out to the cytosol. So there's a 5' end, 3' end. There's polarity there. Triphosphate here, 3' hydroxyl group. We start synthesis, protein synthesis. We start translation. So what happens is the small subunit comes on, then a large subunit. And we start translation. We go from the 5' end to the 3' end. So there is a collinearity. So if we have the RNA sequence, and we know this is where the start codon is, that's going to be amino acid 1, amino acid 2, amino acid 3 from the mRNA. So we don't have to worry about any processing that's already been taking place. So that's collinearity between the sequence within the RNA, mRNA, and the protein. And what happens, we get about 30 amino acids long, and it stalls. There's a slight delay in translation. Translation is a very expensive process. You'll hear more about that. So there's a slight delay. And this slight delay allows that information there within that 30 amino acids to be conveyed. In other words, is that information that says target to the ER. Which form of ER? Rough ER. So that information would then target it to the rough ER if it's going to go through co-translational. So co-translational would be involving the endomembrane system. So that protein can stay within the membrane of the rough ER. It can be in the lumen of the rough ER. But as you know, it then, depending upon the signals that are present, it goes through this processing where it goes to the Golgi, various modifications, and from there it's targeted to the final destination within the endomembrane system. Whether it's going to be in the plasma membrane. So if that protein is destined to reside in the plasmid membrane, that means as it's in the rough ER and as it goes through this Golgi, that protein is going to be embedded in the membrane here. So as we get this vesicle here, there's a topology to this. That is, the two sides of the face of the phospholipid bilayer is not the same, so that when this vesicle emerges from here, it's like this, and when it fuses in exocytosis, what we would see is it's just starting to hit. There's that. Here's this. Eventually it will end up, and I'm going to hatch that so you can keep track of it. We'll end up with something like this. So we maintain that topology, and if we had that protein such that something is exposed here to the cytosolic face, it will always remain in the cytosolic face even here, because there it sticks like that, okay? So we maintain topology as it goes through this. So keep in mind the components of the endomembrane system: plasma membrane, secretion, lysosomes. All right. The other possibility is that instead of being targeted to the rough ER, it could remain in the cytosol, and it would finish translation. So it's fully translated. Okay. Here, that import into rough ER took place while translation was continuing. So from the 30 amino acids long to the final product size, translation's taking process. So that's why that's called co-translational import into the rough ER. This is going to be post, after translation has taken place. And there are some destinations here. So I'd like you to think about that ribosome that has finished translation of that protein. And that protein is now in the cytosol. I'd like you to think right now for a few minutes. There are 5 possible destinations for that. So what are they? This is the exam's Friday, right? You might want to write them down. If you're not sure, talk to your neighbor. You can't do this on Friday, though. All right, that's more than enough time. That's typically about the time frame you'd have on the exam. There's going to be about 50 questions, which means you have to do each question in about a minute. So you can't be struggling for 10 minutes to try to come up with the answer. So some possible destinations are, it could reside in the cytosol, right? How many of you got that? Okay, not even half, more like a third. So it can remain in the cytosol. Certainly lots of proteins remain in the cytosol. Give me some examples of some enzymes that are cytosolic. What's a pathway that we've spent a lot of time on that occurs in the cytosol? Glycolysis. So those glycolytic enzymes have to reside in the cytosol, right? So they would remain there. But we have these other organelles that it could possibly go to. And those are the mitochondria, the chloroplast. I don't know if you noticed, but I said plant cell. So chloroplast. Now, in your figure, it does not list the chloroplast because they did it for an animal cell. So mitochondria, chloroplast, peroxisome, and back to the nucleus through that use of importin. Okay? So any questions about that? Yeah. So the comment is peroxisome co-translation. What did he teach you in lecture? Yeah, it's everything I've always heard has been that the peroxisome is through post-translational. Okay? So anyone else heard otherwise? No? Okay. Okay, never mind. All right. It's okay. Purpose of this is sometimes to clarify those things. That's the beauty of taking practice exams, studying with the partner, is those things where perhaps you don't realize you got it wrong. When you start interacting with others, you'll learn it, okay? So in terms of this, what we see here is a couple graphs, rate versus concentration. I'm trying to illustrate simple versus facilitated diffusion. So if this is a simple diffusion, as you increase the rate, it just keeps increasing. It does not saturate. Facilitated diffusion would saturate at some point. That means that the facilitating molecules, the proteins, whether it's a carrier or channel, gets saturated. It's working as fast as possible. So even though you add more substrate, it's not going to change the rate per carrier per channel, okay? Now, I want to point out a few things. Simple is sometimes called free diffusion. I actually prefer that term, but I don't want you to think free diffusion means there's no energy source. So in fact, figure 73 says that simple and facility diffusion do not require energy, but active transport does require energy. That is definitely wrong. So think about why that is wrong. Why is that statement wrong? There's all sorts of mistakes in books. Why is that statement wrong that this does not require energy? And this one does. And that's true. This one certainly does. But why is it wrong when it says those do not require energy? Sure, there's potential energy, right? Remember, energy, the ability to do work, exists as both potential and kinetic energy. And for free diffusion to occur, we have to have more of Y here than here. So that's potential energy. Okay? For the carrier to work, we have to have more of Q here than there. For the channel to work, we have to more of K here than there. So these do require energy. It's the form of potential energy. Otherwise, you couldn't do work, right? What this is trying to emphasize is that active transport requires the energy in the form of a phosphorylated molecule. Whether it's ATP or GTP or some other form, there's an expenditure of a phosphorylated organic compound. Comment. It won't be an issue on the exam, okay? I will make sure it's clear on the exam, okay? So the question was, if it's asked on the exam, should we answer it as not requiring energy or requiring energy? Is that what the question is going to be? It always requires energy. So on the exam, if a question is like this on the exam, it will make sure that you realize that active transport requires the expenditure of this, that it can work against a gradient. But can these work against a gradient? No. So the type of question, if it was on the exam, would be asking about how can it work with a gradient, how can you overcome a gradient, that sort of thing, okay? Any other questions about that? The next slide here is a graph of reaction profile. You, of course, are very familiar with this. Realized change in energy of activation to overcome that reaction, uncatalyzed versus catalyzed. Okay? It's an important graph. You should be familiar with it. I'd like you to think about this right now. This is pH 4, that's pH 8. We have a compartment. That compartment is established by a membrane. That membrane is selectively permeable. What that means is only certain things can cross it through free diffusion. Things like protons cannot, because protons are going to have lots of water molecules surrounding them to hydrate that positive charge. And as a result, that proton with lots of water molecules around it is not going to go through via free or simple diffusion. So right now, I'd like you to think about this. Where is the gradient, and how would you do it? How could you capture the energy of that gradient? So on your piece of paper right now, be active. Write where more protons are, draw the direction of movement, and then how you could capture that energy. GSI specifically begged me last night, please go over this. They just don't get it. So that's why I'm going over it now, okay? They begged me. Okay, let's quick do this. So pH 4 versus 8, which compartment has more protons? The outside or inside the lumen? pH 4. So that means that the potential energy, the gradient, exists from this location to that. So what we have to do is somehow move those protons from there to there. Since it's a proton, it's not going to cross the membrane freely. So while we move it from here to here, what does it have to go through? A carrier or a channel, in this case, we could couple it to the movement to make ATP. So in fact, that introduces the concept of chemiosmosis. We have more protons in one location versus the other. It takes energy to make that gradient. In photosynthesis, where is the energy that makes that gradient? Where is it located? In photosynthesis, where does the energy come from? Light. And in oxidative phosphorylation, where does the energy come from? Those organic molecules. In this case, the reduced forms NADH and what else? FADH2. So did you have a question? So the question is, ATP synthase a pump or a channel? So it looks like this, which means that in this case, where we make the ATP end, it's going to be the end that has the bulb like this, okay? So we can't go through it freely, so we have to go through that. So the question was, is it a pump or a channel? Most people would consider it to be a pump, although many people say you shouldn't consider it to be a pump, because a pump implies you're going from, a low to a high. In this case, it's high to low. But yes, it involves proteins. It's certainly facilitated in that, and this is what I don't like, active transport is really a subset of facilitation, because facilitation means there's something helping it move. Protein, 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 okay? All right. So yeah, most people consider it to be a pump, okay? To be a carrier in that sense, not a channel, okay? Yes? So you want to move water from the outside in? Is that what you want to do? Okay, so osmosis is the movement of water from a high concentration to a lower concentration, right? So we've talked nothing about the tonicity here, right? We've talked nothing about tonicity. So I think your comment you're trying to get is, if I have more protons here versus here, have I changed the tonicity? Is that what you're trying to get at? You have changed the tonicity, but the change in tonicity is extremely small, okay? Because going from pH 4 versus 8, this is 1 times 10 to the minus 4 moles per liter. And this is 1 times 10 to the minus 8 moles per liter. The volume here is extremely small, so you're probably talking about, maybe a difference of 4,000, 6,000. I'd have to do the math. It's fourfold, so it'd be about 10,000-fold difference. In protons, it's not much, okay? What really is contributing tonicity are all those other ions, okay? But in truth is, yes, you could potentially have to worry about water moving in a little bit more than out, but it's not a significant factor, okay? So, okay. So here, moving of protons is not across the membrane. It has to be facilitated. How? In this case, ATP synthase. You can use it for work, and you need that ability to capture that. Okay, now here's a slide. I want you to think about this. First, you should know what those letters stand for. This is the one that GSI has begged me to do, okay? I introduced it first with that one. You should write the definition of those, what it stands for, and then compare and contrast each of the three of those. Name at least 2 similarities and differences between each. Now, this is not the type of question you would have on the exam, because it's not going to be a short answer. But is the type of question you should be asking yourself now to make sure you understand it, okay? And I'm going to call on people randomly from the audience. It increases participation. Write these down, because you're going to need to exchange your answers with the person next to you, and then you're going to need to hand in that paper, okay? So you need to write this down on a piece of paper so you can exchange it with your neighbor. You're going to then put your name on it and pass it in. What? Write these similarities and differences down on a piece of paper. In case you're not sure, papers like this, right? You know that. Write it down, put your name on it, and then you're going to exchange it with your neighbor. And then you're going to turn it in. And those of you in Webland, you've lost your opportunity. Okay, time's up. Exchange your paper with someone next to you. Read their answers. And you can do three-way exchanges. Just make sure you keep track of them. So go literally exchange them with the next person and read their answer. People are just getting around to exchanging. Okay, time's up. Put your pencils down. Put the piece of paper down. I'm trying to illustrate a couple things here. First off, before I had you pull out the piece of paper, a large number of you were not even doing it, okay? So I asked you to do it on paper just to make you a little bit more active. The other thing is it's too easy in your head to say, yeah, I think I get this, I get this. Having to write it down forces you to formulate a response. It forces you to think it through, okay? Now, a question. Raise your hand if you came up with maybe another answer or an explanation or some aspect of it that you hadn't thought of before by looking at the other person's paper. Okay, so there are hands going up right now. What that illustrates is the exchange of information between each of you in terms of studying that is extremely useful process. Now, this happened in the span of 2 minutes. So you can imagine if you were in study group where you were actively thinking about it, talking about it, going up to a board, discussing it, it would be much, much, much more useful. I'm really on a drive to try to get students to be very active learners. In the end, if you're an active learner, you will spend less time learning than you will as a passive learner. So in the end, the objective is you have to spend less time studying, okay? So a few things here. Substrate level phosphorylation. Key thing here is you should know that it stands for substrate level phosphorylation. And what this does, it involves in phosphorylated, okay, exchange your papers back. And when you get your papers back, put your name on it and your section number. The key thing is name and section number. Because for the exam on Monday morning, so if I could please have the board projected now, because this is important. So if we could switch the camera, thank you, excellent. So it is 8 a.m., that is in the morning. I get lots of students occasionally coming to me about 8:15 p.m. saying, oh my God, I thought it was in the evening. It is not in the evening. That's lab exam 1. The lecture exam on Friday is in the morning. Question 1 is extremely important. It's worth the equivalent of about 40 points. On a 100-point exam, that's a biggie. What this is it's the question that says, what version of the exam do I have? There are going to be multiple versions of the exam. If you bubble in the wrong answer, you will be graded with the answer key that corresponds to that. So if you put C and it was A, we're going to be looking for the answers as though you had C. And if you had A, you're probably going to miss a lot of things, okay? So that question 1 is extremely important. Extremely important, okay. You need to know your discussion section number. That's where you go to take the exam and where you will be seated within the room. And you need to bubble that in and bring your SID. I've had several students email me, oh, I lost my SID. Should I frantically go to Cal 1 card and get a new one or whatever? If you don't have your SID for some reason, then bring a photo ID of some sort. So we have positive identification. All right, so any questions about the exam on Friday morning? No? Excellent. All right, yeah, question. So does it start at 8 or 8:10? At 8 o'clock, we want you seated, ready to go, because we're going to start handing out Scantrons then, okay? And then we have to hand out the exams. Because there are multiple versions of the exam, we have them in a specific order. So we're going to hand them out in a specific order. So we need everyone seated when we start handing out those exams. We have to have it started by 8:10 no matter what, because the next class comes in here at 9. So the chemistry students will be barging in. I'll put signs on the door. We still have to collect those exams. So We want you in your seat by 8 o'clock. We will actually start at 8:10. That does not mean, oh, I don't need to get there till 8:12 or 8:13. You need to be there preferably by 8 o'clock ready to go. The last thing you want to do is be running to the exam because you don't know where it is or you're really late or that. So set multiple alarms, call your friends, get your parents to call you, whatever you need to do, okay? Yeah, we won't start at 8, we'll start at 8:10, but we want you seated ready to go by 8, okay? Good question. Any other questions about the exam? And that's just so we can get it started on time and end on time, okay? So substrate level phosphorylation, it involves a phosphorylated organic compound. You've seen this in glycolysis. For example, when we go from 1,3-bis-phosphoglycerate to 3-phosphoglycerate, that was a phosphorylated organic compound, 1,3-bis-phosphoglycerate, so we could form ATP. Now I expect you to know the other end of the arrow. What's the other end here? ADP. Do I have to put plus PI? No, because where's the phosphate coming from? The phosphorylated organic compound. That's why it's called substrate level phosphorylation. It is not involved in an inorganic phosphate. That phosphate is on a phosphorylated organic compound. Another case you saw this was phosphoenol pyruvate to pyruvate with ATP coming out. You saw this one other location. Where is the third location you talked about substrate level phosphorylation? Krebs cycle. And there it's when it goes from succinate to fumarate. You do not need to remember that. It's actually succinyl-CoA. Do not memorize this. To succinate. And a GTP is made, which is equivalent to a ATP. And you might say, wait, I don't see a phosphorylated organic compound because they don't show you all the intermediate steps, but there is an intermediate step where it's succinate phosphate, and you go from succinate phosphate to succinate, and in that case, then you take this GDP in this case and go there. Again, it does not involve an inorganic phosphate. It involves a phosphorylated organic compound. Substrate level phosphorylation involves organic compounds that are already phosphorylated, donating the phosphate to ADP or the equivalent thereof. Any questions about substrate level phosphorylation? Involves enzymes. It does not involve a proton gradient. It does represent potential energy. It represents energy. The energy here is more than here. As we do this conversion from this to this, we capture a portion of that energy here. Okay? Now, with these two, the similarities are we form a proton gradient, right? The source of the energy to make that proton gradient is different. In this case, the source is those reduced carriers, NADH, and the associated proton, and FADH2. Here, the source of the energy is the photons. In all cases, we're forming that gradient, so we have to have a selectively permeable membrane. We have to have that gradient to form the potential energy. In this case, the terminal electron acceptor is oxygen to form water. That's why this is called oxidative phosphorylation. And this is called photophosphorylation because it's the photons that are doing that. In this case, actually, water is serving as the source of the electrons. And we get off the oxygen, and the protons are there, and of course we move protons to the electron transport chain, okay? Questions? All right, so here's the slide here. If I could get the computer, please. This is from your book. Glycolysis, of course, is out in the cytosol, which is why we talked about substrate level phosphorylation. And when we talked about translation, that some proteins remain in the cytosol through the translation process. We go from glucose to pyruvate. We have to expend some ATP, but we get more ATP out. So there's a net yield substrate level phosphorylation. And then we take the pyruvate, we transport that into the mitochondria, into the matrix. And there's this enzyme complex in the pyruvate, in the matrix, that takes and decarboxylates the pyruvate, and then it reduces NAD plus to NADH. What's the name of that enzyme? It's a dehydration reaction, so what's the name of the enzyme that would dehydrogenate pyruvate? Pyruvate dehydrogenase. Okay? So that's the enzyme that does that. We end up getting NADH, which represents energy, which we can then go through that electron transport chain. We then end up with acetyl-CoA. Acetyl-CoA feeds through the Krebs cycle itself. As we go through this Krebs cycle, we're going to be feeding off 3 NADHs and FADH2. Also, 1 ATP per turn through substrate level phosphorylation. That was this aspect here, the succinyl-CoA to succinate, bottom line, substrate level phosphorylation. But we have all this NADH, this FADH2 that's reduced. So in order to capture that energy, we go through the electron transport chain or system, and we make that gradient, and we couple that gradient to the formation of ATP. Here just shows you the locations. Again, if you're struggling to visualize this stuff, make diagrams. It helps tremendously with your learning process. Really, there's a lot of stuff going on here, right? So if you make diagrams, it really helps you to organize this. So this is from a different book. They actually leave off a step here. It's the Campbell book. Older editions included it. The most recent edition, for some reason, just left it out. And so we said pyruvate's formed in the cytosol. That gets transported into the matrix here. The pyruvate gets oxidized, that dehydrogenation reaction, and then acetyl-CoA enters the citric acid cycle. So they completely leave that step off. So we know this forms NADH and FADH2, which goes through that transport chain. Oh my God, what's going on here? To the left, glycolysis. How many of you have memorized those steps? Names of the enzymes. Delta Gs. Number of bonds formed. Number of bonds broken. No one? Excellent. You shouldn't. This stuff here, a lot of steps, 10 steps. You don't need to know each step. But let's focus on the key aspects of this. The key aspects. We start off with glucose. We spend a couple ATP. They call about priming the pump. We form fructose 1,6-bisphosphate. This is a 6-carbon molecule. When it gets broken down into 2 3-carbon molecules. Dihydroxyacetone phosphate, not important for you to remember. Glyceraldehyde 3-phosphate. That is an aldehyde. This is the structure. That's the number 1 carbon, number 2, number 3. So it's 3-phosphoglyceraldehyde. Okay? But this DHAP gets isomerized to this. So we will go through this cycle twice. I'm only showing it once, but remember it goes through it twice. So everything here will have to be doubled at the end of glycolysis. So we start with glyceraldehyde 3-phosphate. It's an aldehyde. We are going to convert that aldehyde to an acid. Is that an oxidation reduction step there? Is the molecule being oxidized or reduced? Being oxidized. Remember oxidation steps? Things like going from CH4 to the alcohol to the aldehyde. So this is the OH to the aldehyde to the acid and ultimately to CO2. So the molecule, the organic molecule, is being oxidized. What must happen to something else? It must be reduced, okay? So what gets reduced? NAD plus to the NADH plus the associated proton. Now, if you notice something here, we have glyceraldehyde 3-phosphate here. We form 1,3-bisphosphoglycerate, okay? So we'll form the acid, but now it's got 2 phosphates on it at different positions. So it's called bis. Still got the 3 there, still got the alcohol there, but now this is an acid. It is now 1,3-bisphosphoglycerate, an acid. This is oxidized relative to that. We had to reduce this. There's our reduced molecule out in the cytosol. Since the electron transport chain is in the inner membrane of the mitochondria, we will eventually have to transport that NADH into the mitochondria to be able to capture the reducing power there, right? It's the reducing power of NADH and FADH2 that powers the electron transfer system, the chain, but ultimately we have an electron acceptor, which is oxygen, okay? Question? Yes, so there are different ways to, so the comment was I've seen the G3P also written as 3PGAL. This is glyceraldehyde 3-phosphate, this is 3-phosphoglyceraldehyde. It's the same molecule, just different ways to abbreviate it, okay? I personally prefer this method to emphasize the aldehyde, but Most books, most literature uses this form, so that's why I used it there, okay? But the key thing is it's an aldehyde, we're going to an acid, all right? And then we have this 1,3-bis-phosphoglycerate. We're going to go to 3-phosphoglycerate. That's this step here. We form an ATP through substrate level phosphorylation. Eventually we get to phosphoenol pyruvate. We go to pyruvate. Again, we form ATP. So we form 2 ATP molecules starting with the 3-phosphoglyceraldehyde, glyceraldehyde 3-phosphate, either one, to pyruvate. But since this is twice, we get 4 of these. So I tried to show red inputs, green outputs. So here we're incorporating that in inorganic phosphate so that later on we can get a phosphorylated organic compound to do the substrate level phosphorylation. Any questions about that? That is key step there. That's the one thing Dr. Malkin has talked about repeatedly about glycolysis. So we get pyruvate. Pyruvate then gets oxidized into that transport in the mitochondria. So as we go in, that pyruvate dehydrogenase in the matrix will reduce NAD plus to the NADH. We release CO2, and we form acetyl. That acetyl gets attached to CoA. We now have acetyl-CoA, and that can then enter the citric acid cycle, Krebs cycle, TCA cycle, various names for it. And as we go through this process, we will take and eventually take that acetyl group, which is like this, that's the acetyl group. We'll eventually oxidize it to CO2. 2 of them. But if you notice, we have hydrogens there. So as a result, we can reduce NAD plus or potentially reduce FAD as we go through this process, okay? All right, so that's that aspect of it. Any questions about that? You should be able to create your own table about inputs and outputs with each step. 4 steps: glycolysis, pyruvate dehydrogenation, the Krebs cycle, and oxidative phosphorylation. Be able to do that by Thursday at noon so that you can repeat it a couple times. So this just shows the electron transport chain. Be familiar with this. Complex 1, NADH is entering here. We're going to take and transfer those hydrogen molecules to ubiquinone. So there's electrons here, but that carries hydrogen, so we grab protons here and we're pumping them. So basically we're going to pump protons from one face of the membrane to the other. We form a gradient. This is in the intermembrane space. So the example I'd put on the board earlier was illustrative of the thylakoid lumen or thylakoid space. This, the protons are being pumped into the intermembrane space. Same thing though, we are making a gradient, which represents potential energy, which we can capture to the powering of the ATP synthase to make ATP. So in this case, it's in the lumen here, the matrix, okay? So we go through from complex 1 to complex 3, complex 4. Eventually, we add electrons and protons to oxygen to form water, okay? So complex 1, complex 3, complex 4. FADH2 enters at complex 2. Again, we pump protons to this process. So we pump fewer protons for FADH2 than we do for NADH. Hence, we will get fewer ATP molecules per FADH2 than we do for NADH because fewer protons are pumped, okay? All right, I see that something happened to my diagram here, and it's missing. What was I doing here? Oh, accounting for the NADHs and FADH2s. Unfortunately, everything on this side's gone. Is it on your printout? Yes, no? It's on your printout, but not on my display. Okay, well, things like that happen, okay? What was going on over here was this was supposed to be showing the NADH from the glycolysis. There's 2 of them, 2 NADHs from glycolysis out in the cytosol. If we didn't have to transport them, we could get 3 ATP. This is Dr. Malkin's accounting. Your textbook uses a different numerical value. Your textbook uses 2.5. For our purposes, we're going to use 3, okay? So for each NADH out here in the cytosol, we get 3. If this was a prokaryote, that NADH is located where? If this was a prokaryote, where would the NADH be? All right, let's just draw a diagram then, since we're not sure. Cell wall, cell membrane. Where do you think the NADH is going to be? It's going to be in the cytosol. Where's the electron transport chain? There's only one place for it to be. It's got to be in a membrane. What's the one membrane? Plasma membrane. So in this case, the NADH does not have to be transported to where the membrane is. So in prokaryotes, there's no cost associated with that NADH from glycolysis because there's no transport required. In a eukaryote, since glycolysis is in the cytosol and we got to get it to the matrix, people sometimes talk about a transport cost. So if there's no transport cost, it's 3 ATP per NADH. There's 2 out there in the cytosol from glycolysis. That makes 6. If we consider transport costs, sometimes people take off 2 ATPs. So that's why you hear sometimes 38 versus 36, okay? If I could get the computer, please. Okay, excellent, thank you. 6 ATP here. These are for the 2 NADHs from pyruvate dehydrogenation. Those 2 NADHs are directly in the matrix or in the cytosol if we're talking about the prokaryote, but let's just continue with the eukaryotes. So those 2 NADHs, remember 2 cycles, yield 6 ATP. From the Krebs cycle, we get 3 NADHs per turn. Since we do it twice, we get 6 NADHs, again, already in the matrix, so we get 6 times 3 for 18. And then we have FADH 2 per turn. We have 2. That makes 2 ATP per each, yields 4. There's 34 there, but we had 2 ATP, substrate level phosphorylation, net during glycolysis, and 2 turns of the Krebs cycle, so 2 ATPs through substrate level phosphorylation, so it's 34. In red represents oxidative phosphorylation. The 4 through substrate, that's 38. And again, if we have to expend 2 ATPs for transport, that's why you sometimes see the value of 36. Your textbook uses different numbers. They use 2.5, so I wanted to make that clarification. Any questions about this? In fact, your textbook uses 2.5, whether it's NADH or FADH2. We're using 3 for NADH, 2 for FA. Oh, there it comes. All right, so yeah, so that just shows, there's your textbook doing 2.5. We're doing the counting of 3, 3, 3, 2, and taking into account transport costs. For photosynthesis, remember that the light reactions occur in the thylakoid membrane. That's where we're going to be making that proton gradient, so there's going to be more protons in the lumen here. And what we'll be doing is, as those protons move out, we make ATP in the stroma, and we make NADPH in the stroma, so we can do a Calvin cycle. Okay? And you should be just familiar with those photosystem 2, photosystem 1, and the carriers. I want to actually move forward a little bit here to where students tend to have more difficulty, and that is through the Calvin cycle in glycolysis, because people really struggle with this. So we just did glycolysis earlier. We said we had 3-phosphoglyceraldehyde. We go to 1,3-bisphosphoglycerate. We incorporate the inorganic phosphate. We get the reduced NADH. And then we go from 1,3-bisphosphoglycerate to 3-phosphoglycerate, form ATP. That was, again, on this board here. This is that bisphosphoglycerate to the 3-phosphoglycerate, capturing that phosphate group. So notice, in this direction, we are getting out NADH, associated proton, and ATP. So if we are going to, in photosynthesis, do the reverse of that, we have 1,5-ribulose bisphosphate carboxylase incorporating a CO2. It forms a 6-carbon molecule, very, very, very short-lived. That gets broken down into 2 molecules of 3-phosphoglycerate. Notice the similarities. Those are identical molecules. So what's happening in the Calvin cycle is we got to spend ATP to form the 1,3-bisphosphoglycerate. You've seen that before. Then we go to 3-phosphoglyceraldehyde. If we get NADH out here, what do we have to spend here? We have to spend a reduced molecule, and we will be getting out an inorganic phosphate. So basically, you're familiar with glycolysis. Most students get this. Realize that Calvin cycle is pretty much kind of the opposite of it. That's why we have to spend ATP and NADPH. We'll spend 6 ATP and 6 NADPHs to eventually incorporate 3 CO2s. Okay? This shows you sort of the balance. So if we have 5 carbons, 3 CO2s, 6 3 carbons, 6 of these, have to spend 6 ATP. Go to 6 of those, we got to spend 6 of these, we get off 6 inorganic phosphates. The bottom line in this, we're going to be spending 6 ATP and 6 NADPH. We'll end up with 6 3-phosphoglyceraldehydes, One can be funneled off to make sugars and be synthetic and all that sort of stuff. But to continue the process, we have to regenerate these 3 molecules of 1,5-ribulose bisphosphate. So in the process of this, we're going to spend 3 ATPs. It's an extremely complicated set of reactions. You form 10 carbon molecules, things like that, blah, blah, blah. It's really complicated. We don't need to know it. All we need to know is we go from 3 carbons to 3 5 carbons, and it takes 3 ATP. All right? Okay? I posted these slides on BSpace. So everything's on BSpace, okay? So we're going to move on. That's the complicated stuff. We already saw the big picture there. In terms of CAM versus C4 plants, I actually really dislike the name C4 plants because it's a little misleading. I really think they should be called C7, but I didn't get into the naming. This was before my time. The reason why I don't like the term C4 is C4 and CAM plants are referring to the initial capturing of the carbon dioxide. Both CAM and C4 and C3 plants all ultimately have to do the Calvin cycle. They all ultimately have to do the Calvin cycle. So where they differ is in C3 plants, you have that RUBP capturing the CO2, and we form those 2 3 carbon molecules. That's C3 plants. In C4 plants, what we do is we use phosphoenolpyruvate to capture the CO2 to form a 4 carbon molecule, and eventually that 4 carbon molecule will be released to give the CO2 to form a 3 carbon molecule, which then gets converted back to that, and it takes ATP. So a C4 plant ultimately ends up doing the Calvin cycle as well. So it's kind of C4 plus C3, right, in a sense. With a CAM plant, and to do this, it is spatially separated. So this process is occurring out in the mesophyll cell. So it's a spatial solution, and this process is occurring in the bundle sheath cell. The key thing here is that since we are using PEP carboxylase, it has no oxygenase activity. So we don't have to worry about oxygen competition here because the enzymes doesn't treat it as a substrate. When we get to the Calvin cycle, since we use an organic acid here, when we decarboxylate this, we actually can get very high levels of CO2 relative to just being dissolved in the liquid because we have an organic acid that's donating it. So what we do here is where the Calvin cycle is operating, we increase the ratio of CO2 to oxygen. Based upon Le Chatelier principle, you know if you increase one substrate relative to another, you will increase that reaction relative to that. In CAM plants, they do a similar thing where they use PEP carboxylase in that. They capture that C4 acid. They go through this process. Then they go through the CO2 as well. So everything's similar except this is nighttime. It's a temporal solution. During the nighttime, they are capturing the CO2, so you minimize water loss because it's usually cooler at night. They capture the CO2. They form the C4 acid. It's in the vacuole, very, very high concentrations. During the daytime, they release the CO2, and they drive the reaction. So the reason why this has to occur, this part, at daytime, why they have to do the actual Calvin cycle during the daytime, is because that's when you have your ATP and your NADPH from the light reactions. You can't make enough of those molecules. So what you do is you take, so it's a spatial versus temporal solution, okay? So that's it. Good luck on the exam on Friday. I'll have office hours from 12 to 3 today.